Hi, in this presentation we will discuss about biosimilars, a simplified overview. Biosimilars are just like generics. In case of chemical drugs, the version is called generics, but in case of biological drugs, the generic version is called biosimilars. It is not called generic, it is called biosimilar because biological drugs are very complex as compared to chemical drugs. They need huge amount of money, huge time, huge amount of data to develop biologics, and to develop the generic version of biologic or biosimilars, that also requires huge amount of data, huge amount of time and money. And when you compare it with the chemical drug, for developing a generic version of a chemical drug, you need just $1 to $5 million, 3 to 5 years. But in case of biosimilars, you need $100 to $200 million and 8 to 10 years to develop a biosimilar. That's why the generic version of biologics is called biosimilar and not generic. So, a biosimilar is a biological medicinal product that contain a version of already authorized original biological medicinal product, which is called reference biological product. A biosimilar demonstrates similarity to reference biological product in terms of quality characteristics, biological activity, and safety and efficacy profile based on a comprehensive comparability exercise. Different countries they use different terms for biosimilars. Like in USA, it is called biosimilar. Australia, biosimilar medicinal products. In Taiwan and Korea, biosimilar biological products. In Japan, they're called follow-on biologics. In China, non-innovator biological products. In India, called similar biologics. In Europe, called similar biological medicinal products. WHO call them similar biotherapeutic products. In Singapore, it is called similar biological products, and in Canada, it is called subsequent entry biologics. So there are different benefits and cost saving in case of biosimilars. Availability of biosimilar medicinal product, they enhance competition. They can improve patient access to biological medicines and can contribute to the financial sustainability of healthcare system because biological products they are very costly, and the generic version or biosimilar are usually priced slightly lower than the price of the original biological products. The improved affordability of healthcare system that could result from use of biosimilar is real, and it has been proven in many countries. With the introduction of biosimilars, there was huge cost saving in the healthcare system. Biosimilar medicine product can be expected to be offered at a price lower than the reference biological product, partly because of production process efficiencies, partly because of reduced cost of a streamlined development program, as well as result of competition. Greatest cost saving are likely to result from clinical trial program, since biosimilar would require lesser clinical data to support its approval. Similar biosimilar development costs, the European Generic Association has estimated between 100 and $150 million and 8 to 10 years for developing a biosimilar. The US Federal Trade Commission have estimated cost between 100 and 200 million for developing a biosimilar product. When you compare with a chemical drug, for developing generic uh, version of a chemical drug, it costs between $1 to $5 million only and takes around 3 to 5 years. It is imperative for biosimilar manufacturer to minimize the cost. whenever possible, and greatest saving are likely to result from clinical trial program because biosimilar would require lesser clinical data for its approval. And a phase 1 3-arm trial is one avenue by which it can be done. It can accelerate approval process in US and Europe. As per WHO, the key principles for approval of biosimilar are: development of a biosimilar involves stepwise comparability exercise, starting with the comparison of quality characteristics of biosimilar with the reference biological product. After each step of comparability exercise, decisions should proceed further with development of biosimilars should be evaluated. Demonstration of similarity of a biosimilar and reference product in terms of quality is prerequisite for reduction of non-clinical clinical data requirements. Basis for licensing a product as biosimilar depends on demonstration of similarity to a suitable reference biological product in terms of quality, non-clinical and clinical parameters. Biosimilars are not generic medicines, and many characteristics associated with authorization process they generally do not apply. If relevant differences are found in quality, non-clinical and clinical studies, then product will not likely qualify as a biosimilar, and a more extensive non-clinical and clinical data set will likely be required to support this application for licensing. Biosimilars, like other biotherapeutic products, they require effective regulatory oversight for management of their potential risks and in order to maximize their benefits. The decision to license a product as biosimilar should be based on evaluation of whole data package for each of these mentioned parameters. Biosimilar development is a stepwise process or stepwise approach. One should apply stepwise approach to data generation and should evaluate the residual uncertainty after each step. First, one should compare analytical data of biosimilar and reference biological product, then do the animal studies, comparative animal studies, then comparative PKPD study between biosimilar and reference biological product, and then one need to do clinical immunogenicity assessment. And if required, if there are residual uncertainty after these steps, then additional clinical study may be required. So biosimilar, the stepwise development process starts with the comparative exercise comparing the analytical or quality data of biosimilar with the reference product in terms of manufacturing process, product characterization, structural and physical chemical properties, biological activity, immunological properties, purity and impurity profile, and stability data. The next step is non-clinical studies comparison. So one has to do the comparative studies of biosimilar with reference biological product in terms of both in vivo and in vitro studies. Then after animal study, one has to do the clinical studies, and which again should be comparative between biosimilar and reference product. One has to do the PK studies, pharmacodynamic studies, confirmatory safety and efficacy studies, and required to do the immunogenicity study and safety studies in order to generate the immunogenicity data and safety data. So amount of studies required will all depends on amount of uncertainty remains after each step in the development process. So in this slide, you can see the comparison of data requirement for developing a biosimilar product and a reference biological original product. On left side, you can see you will need to submit the analytical data, non-clinical data, clinical pharmacology data and additional clinical studies data in case of reference biological original product. And in case of biosimilar product data package, you need to do huge amount of analytical data comparison of biosimilar and reference biological product. You will need some amount of non-clinical data also, and you will need at least one PKPD or clinical pharmacology studies. And you may require to do additional clinical studies if there are still remaining uncertainties after analytical, non-clinical, and clinical pharmacological studies. So this is a multiple step approach to demonstrate biosimilarity, and quality is a foundation in terms of analytical data. So in case of biosimilar, when you compare with the original data package of reference biological product, you will need larger amount of analytical data and lesser amount of non-clinical and clinical data. So the analytical quality comparison characterization, you will need to do the comparability exercise between biological biological product, reference product, and biosimilar product. So one should include the comprehensive side-by-side analysis of biosimilar and reference product using sensitive and orthogonal method to determine not only the similarities but also the potential differences in quality attributes. Any differences detected in quality attributes will have to be appropriately justified with regard to their potential impact on safety and efficacy. So one has to do the comparative analytical studies. One has to one has to consider the comparison of analytical data. One has to do the comparative analytical or quality studies in terms of analytical consideration, physicochemical properties, biological activities, immunochemical properties, specification, purity and impurity profiles. So one should do the comparative quality or analytical comparison between biosimilar and reference product. Next step is non-clinical studies requirement. So one has to do comparative in vitro and in vivo studies. In vitro studies may involve receptor binding studies, binding to antigen or enzyme, and these in vitro studies should be comparative. One should access the concentration, activity, and binding relationship in these studies. In vivo studies may be PKPD studies, which again should be comparative or biosimilar and reference product, and which will include concentration response assessment. A repeat dose toxicity study with refined design or an in-life evaluation of safety parameter may be considered. Studies regarding safety pharmacology, reproduction toxicology, carcinogenicity studies are usually not required for biosimilar. Studies on local tolerance are usually not required, but if excipient is new for the intended clinical route, then local tolerance studies may be required. Now, biosimilars clinical studies requirements. So one has to do the comparative PK studies first. So comparative PK studies are essential part of biosimilar development program. Design of study depends on various factors including clinical context, safety, PK characteristics of the reference product. Then one has to do the PD or pharmacodynamic comparative studies of biosimilar and reference product. PD markers should be added to the pharmacokinetic studies whenever possible, and PD markers should be selected on basis of their clinical relevance. Normally, comparative efficacy trials are required for demonstration of clinical comparability, but in certain cases, comparative PKPD studies between test and reference product may be sufficient to demonstrate the comparability clinically. When evidence to establish clinical comparability will be derived from studies with PD markers or biomarkers, it it is recommended to discuss such approach with the regulatory authorities. Efficacy trials, which again should be comparative trials between biosimilar and reference biologic product. Usually, it is necessary to demonstrate comparable clinical efficacy of biosimilar and reference medicine product in adequately powered, randomized, parallel group comparative clinical trials, preferably double blind. In general, an equivalence design should be used. Study population should be representative of approved therapeutic indication. Use of non-inferiority design may be acceptable if justified on basis of strong scientific rationale. It is recommended to discuss use of non-inferiority design with the regulatory authority well in advance. Clinical safety data again should be comparative. Clinical safety is important throughout the clinical development program and is captured during initial PKPD evaluation and also as a part of potential clinical efficacy study establishing comparability. So safety data are collected from all PKPD studies and efficacy studies. Potential for immunogenicity of a biosimilar should always be investigated in comparative manner with the reference product. For biological product with multiple indication, immunogenicity could differ among indication, and absence of immunogenicity assessment in particular indication of biosimilar may have to be justified. Now coming to the requirement of clinical studies data in US. In US, the biosimilar application is called 351K application, and which must include the clinical study or studies, including an assessment of immunogenicity and PKPD, sufficient to demonstrate safety, purity, and potency. The scope and magnitude of clinical studies will depend on context of residual uncertainty in quality or analytical data comparison and non-clinical studies. Lessening number or narrowing the scope of any of these types of clinical studies, that is PKPD or clinical immunogenicity and safety effectiveness studies, should be scientifically justified. If you want to submit lesser amount of data, you should submit the justification. Human PKPD studies should be comparative. Type of pharmacology studies will depend on residual uncertainties about biosimilar that these studies are capable of addressing. FDA recommends use of crossover design for product with shorter half-life, and for product with longer half-life, more than 5 days, the parallel studies will usually be needed. One should include PD endpoints and pharmacometric analysis. In certain circumstances, PKPD data may provide sufficient clinical data to support demonstration of biosimilarities. So in case of the simple biological product, only PKPD data may be sufficient to support the demonstration of biosimilarities. But that will all depend on type of biological product. And sponsors are encouraged to discuss crucial aspect of the clinical pharmacology development plan with the FDA in early stage of development. Clinical immunogenicity assessment, at least one comparative clinical studies on immunogenicity with a reference product will generally be required. FDA recommends use of comparative parallel design, that is head-to-head study. Safety and immunogenicity data from clinical pharmacology studies should be collected and evaluated. Clinical safety and effectiveness data. Comparative safety and effectiveness data will be necessary to support demonstration of biosimilarity if there are residual uncertainties about biosimilarity of two products based on structural functional characterization, animal testing, human PKPD data, and clinical immunogenicity assessment. Sponsor may provide scientific justification if it believes that some of or all of the comparison on clinical safety and effectiveness are not necessary. Clinical studies should be designed such that they can demonstrate that proposed product has neither decrease or nor increase activity compared to reference product. So you have to demonstrate the similarity, not the inferiority or superiority of biological with the reference product. A study implying the two-sided test, that is an equivalence design, is more straightforward design. But in some cases, one-sided test that is non-inferiority design may be appropriate for comparing safety and effectiveness. But selection of design should be discussed well in advance with the FDA. Now coming to extrapolation of indications. In case of reference medicinal product, if it is having more than one indication, then efficacy and safety of biosimilar is to be justified and if necessary demonstrated separately for each of the indications. And justification will depend on clinical experience, available literature, mechanism of action of biological in each indication, including its degree of certainty and on receptor type of receptors involved in each indication. If there is evidence that different active sites of the reference product, different receptors of the targets are involved in different indications, or if the safety profile of the product differs in therapeutic indication, then additional data may be needed to justify extrapolation of safety and efficacy from indication studies in pivotal clinical trials. The extent of such data should be considered in light of totality of evidence derived from biosimilar comparative exercise and potential remaining uncertainties. Coming to reference product requirement, which product you will consider as a reference for comparison with your biosimilar. Reference medicine product must be a medicinal product authorized in that region. Like in case of Europe, the reference medicinal product or original medicinal biological product should be approved in Europe on the basis of complete dossier. However, with the aim of facilitating global development of biosimilars and to avoid unnecessary repetition of clinical trial, it may be possible for an applicant to compare biosimilar in certain clinical studies and in vivo non-clinical studies with a non-European comparator or reference product. That is the reference product which is not authorized in Europe. Maybe sometime the company is required to do the clinical trials in US instead of Europe. So in that country, they may be comparing with the US reference product. So if they are doing that, then applicants should provide adequate data and information to justify the relevance of the comparative data, and they need to establish an acceptable bridge to a Europe authorized reference product. In that case, they have to do a separate bridging study in which they will be comparing. They will be doing three-arm study. In one arm they will be taking biosimilar. In second arm they will be taking the European biological reference product, and in third arm they will be taking the non-European reference product. So that will be three-arm bridging studies, and in that three-arm bridging study they have to prove that the efficacy and safety of all three biosimilar, European reference and non-European reference should be same. So overall acceptability of such an approach and type of bridging data will be decided on case by case basis, and it should be discussed with the regulatory authorities. And the final determination about adequacy of scientific justification and bridge will be made during the review of application. And same principle applies in the US. If the study is done outside US, then one has to do the bridging study in US comparing biosimilar US reference product and non-US reference product. Now, pharmacovigilance for biosimilars. Data from pre-authorization clinical studies are usually insufficient to identify rare side effects. Therefore, clinical safety of biosimilar must be monitored closely on an ongoing basis during post-approval phase, including continued benefit risk assessment. Within authorization procedure, applicants should present a description of pharmacovigilance system and a risk management plan in accordance with the legislation or regulatory guidelines. Risk management plans should take into account identified and potential risks associated with the use of reference product, and if applicable, additional potential risk identified during development program of biosimilar, and should entail how these issues will be addressed in post-marketing follow-up. Immunogenicity should be specifically addressed. Within pharmacovigilance plans, any specific safety monitoring imposed for reference product or product class should be taken into consideration. Applicants are encouraged to participate in already existing pharmacoepidemiological studies in place for reference product. Risk minimization activities in place for reference medicine products should also be included in the management program of biosimilar. So that's all in this presentation. Thank you very much.